Good evening, everyone. I am your biochemistry mentor, Dr. Pooja. And since you people have your NEET MDS exam day after, so this is just a brief video. This is just a brief attempt from my side. I will be discussing those questions which have been asked in, previous year questions paper, right? So let's go for it's a kind of recall session also because these questions have been asked in previous years. So it is NEET MDS Recall 2019. I am Dr. Pooja. So if you have any queries regarding biochemistry, you can always log into my website, which is biochemistrysimplified.com. And if you like my video, don't forget to subscribe to my channel because I keep on updating the questions which have been asked in your, competitive exams. So let's start up with the discussion. Question number 1. Histones are rich in what type of amino acids? Now, first of all, you should have a brief knowledge of what do you mean by histones. Now, basically, these histones are the basic proteins which wrap themselves, or you can say which takes DNA as a wrapping one. And these histones are of four main types: H2A, H2B, H3, H4. H1 acts as a linker DNA, right? And these histones are rich in basic amino acids like lysine and arginine. And they play a very, very important role in transcription. They help in regulating transcription, or they help in switching on and off of genes. Very, very important. Switching on and switching off. So if we see how DNA is wrapped around histones, I'll just give you a brief idea of this. As I told you, H2A, H2B, H3, H4. They are present as an octamer. So 5, 6, 7, 8. So this is what is an octamer, in which DNA is wrapped like this. So when DNA is wrapped around this octamer, it is known as a nucleosome. Right. So this nucleosome is actually present on the chromonimeta or thread-like structure like this, and H1 acts as a linker DNA. So when DNA is wrapped around histones, obviously DNA cannot express itself, cannot undergo transcription, so it's kind of switching off. And when they require, the function of DNA is there, automatically reverse of this happens. DNA is exposed and switching on of genes is there, which means transcription and translation will be there, right? So this is a very simple question. Histones are rich in basic amino acids, which are lysine and arginine. Moving on to the second question, mitochondria are rich in which enzyme of gluconeogenesis? Now, this is a very important question which was asked in last year exam. And this year, somewhat related to this can be asked. Gluconeogenesis is the process of formation of glucose from non-carbohydrate precursors, which include lactic acid, glycerol, and other amino acids. But it is also a reciprocal of glycolysis. So it's like glycolysis में the three steps which are made irreversible in glycolysis are made reversible in gluconeogenesis. Now, if you look at the enzymes, and this gluconeogenesis occurs in two organelles, mitochondria and cytoplasm. Right. So if we look at the enzymes, see what happens is first reaction takes place in the mitochondria. After that, it starts with the cytoplasm. So if you see the reactions, if we compare glycolysis, the last step of glycolysis, which is PEP to pyruvic acid, here pyruvic acid will be converted into PEP. So this is a two-step reaction. What happens is this pyruvic acid is already present in the mitochondria. Pyruvic acid and PEP. So this pyruvic acid is converted into oxaloacetic acid in the presence of enzyme pyruvate carboxylase. On the other hand, this pyruvic acid is also being converted into acetyl coenzyme A, where this TCA cycle is going. Right? So you can say pyruvate carboxylase, the enzyme present in mitochondria. Then this oxaloacetic acid is converted into malic acid, as oxaloacetic acid is impermeable to mitochondrial membrane. It will be converted into malic acid. Malic acid will be transported here, where it will be converted into oxaloacetic acid, and then PEP will be produced. So this reaction takes place in the cytosol. So overall, if we see this, all the steps of gluconeogenesis, only pyruvate carboxylase is the one which is taking place in the mitochondria. Right, so let's see the question. Glucose 6-phosphatase, it's not present in mitochondria. Fructose is again in the cytosol. Pyruvate dehydrogenase, it is not an enzyme of gluconeogenesis. So obviously the answer would be pyruvate carboxylase, which is the only enzyme present in the mitochondria. Right. Now here I am going to tell you one mnemonic also. There, always remember there is a mnemonic called HUG. You won't forget this one, HUG. So HUG में H stands for heme synthesis, U stands for urea cycle, and this G stands for gluconeogenesis. So these are the three processes which always take part in two organelles, mitochondria and cytoplasm. Okay, so I hope this question is clear to all of you. The answer will be D. Moving over to the next question, true regarding carbohydrates. Now this is again a question asked last year. Milk-derived artificial sugars increase the risk of cancer. It's not true for carbohydrates. Lactose and maltose are disaccharides. Yes, they are disaccharides. Sugars with high glycemic index increases the risk of type 1 diabetes. Now, if we talk about type 1 diabetes, it is not associated with this high glycemic index. It doesn't have a direct influence. High glycemic index, yes, it has a direct link for type 2 diabetes, not type 1. Then de novo carbohydrate synthesis cannot occur from glycerol and protein metabolism products. So if we talk about de novo, it's not de novo. The synthesis of carbohydrates is never de novo, because whatever is synthesized, de novo means ki if we have to make a 6-member compound, we have to start from 1 or 2. So de novo pathway is actually for fats or lipogenesis and in purine synthesis. Right. So overall, if we see the options, true regarding carbohydrates would be B. And you know that lactose and maltose are disaccharides. This lactose is produced from beta glucose plus beta galactose, disaccharide, and maltose is produced from alpha glucose plus alpha glucose. So the answer would be this. I hope it is clear. Moving on to the next question. I have seen so many students asking me this question so many times. They have, I don't know why there is so much of confusion regarding uncoiling of DNA. See, every enzyme has its own function to perform. If we see enzymes which cause uncoiling of DNA, see, because DNA is double-stranded, we have to see where lies the origin of replication. When we know the origin of replication is there, we get that replication fork. So the enzyme responsible for uncoiling of DNA is helicases. Right. If we look at other enzymes, topoisomerases is the stress relieving enzyme. Stress relieving enzyme, because DNA is double stranded, it requires, it is so tightly wound that so much of stress will be generated when we try to pull DNA strands apart. So topoisomerases is actually relieving the stress and strain which is caused in that double helix molecule, just to make that unwinding easy. Right? So topoisomerases is a stress relieving enzyme, but helicases is a uncoiling enzyme, या unwinding enzyme, along with SSB, single strand binding proteins. DNA ligase is known as glue of DNA. Glue of DNA in the sense it tries to stick DNA together during either repair mechanism or during the generation and synthesis of the primers. Or fragments, you can say, because Okazaki fragments in the end are sealed by this DNA ligase. And restriction endonucleases, all of you know that they are known as the molecular scissors, because they cut DNA at specific sites. So the answer to this would be simple helicases, or C. Right? I hope this is clear to all of you again. Then another question is, which of the following is a hemoprotein? Now, this is a classification of proteins. If we have to classify proteins, they are simple, conjugate, and derived. Right. Simple means they are the simple proteins. They can only be hydrolyzed further. Conjugate means they have one protein part, along with this some other non-protein part is associated. So this heme protein will come under conjugate protein, or it is also known as chromoprotein, color protein, right? So it has to be myoglobin, right? If we look at casein, casein is the milk protein, or it is a phosphoprotein. Albumin is a simple protein. Right. So the answer to this question is hemoprotein. The example is myoglobin. I hope this question is also clear to you. Then we move over to the next. Base stacking of DNA is characterized by. Now, base stacking, what do you mean by the term base stacking? It is actually, because whenever we have to study the absorption of DNA, whenever we have to quantify DNA, we can quantify only on the amount of the bases present, isn't it? So if we look at the bases, guanine is triple bond with cytosine, adenine forms a double bond with thymine. So, what happens here is, if we have to see the chromicity, or you can say on the basis of the number of bonds, if we see guanine and cytosine will have triple bond, so they will require more energy to break than adenine and thymine, isn't it? So they are going to absorb a particular reagent, and absorbance is studied at 260 nanometers. Right, we read the absorbance of DNA at 260 nanometers, and we say if DNA has more GC base pairs, it will absorb more color, so it will lead to hyperchromicity. Then DNA which has 80 base pairs, so it will lead to hypochromicity. So what we say is that base taking of DNA will be characterized by the more number of GC base pairs. And if we say GC base pairs, so it has to be hyperchromicity. I hope it is clear. Because base stacking, and always remember that DNA absorbs at 260 nanometers, or it is UV range. Okay, so I hope this question is clear to you. We can quantify DNA at 260 nanometers depending upon the number of bases. Moving on to the next question, amino acid excreted in the urine in cystinosis. Now cystinosis is simple cystinuria, and there is excretion of 4 amino acids in urine, which is obviously cysteine, ornithine, lysine, and arginine. Right. So there is defective reabsorption of these in cystinosis. So obviously this is also known as cola or coal. So you can see cysteine, ornithine, arginine, and lysine. So the answer would be all of the above. So the amino acids excreted in urine in cystinosis would be all the above. Then next question, during conversion of glycerol to pyruvic acid, the first glycolytic intermediate to form is. Now, this is simple glycolysis. And glycolysis, mein, if we see, I'll just write after the splitting step, because all the 4 intermediates are after the splitting step. So you can say aldolase gets differentiated between 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. This glyceraldehyde will be converted into 1,3-bisphosphoglyceric acid, and this 1,3-bisphosphoglyceric acid will be converted into 3-phosphoglyceric acid. So the difference between these two is aldehyde and acid, and then 2-PGA. So if we see the chronological order here, during conversion of glycerol to pyruvic acid, the first intermediate. So if we look at the first intermediate which is produced, it has to be glycerol, the first glycolytic intermediate. So it has to be 3-phosphoglyceraldehyde, which is an isomer of dihydroxyacetone phosphate. And after that, you can say 3-PGA, 2-PGA, they will be produced. Right? So the answer would be C. Moving on to the next question. This question was again asked in 2019. All of the following are essential amino acids except. Now, if we look at the concept of essential amino acids are the one which cannot be synthesized. They have to be provided from outside diet. So if we look at all the 3, there are essential, there are semi-essential. Okay, if we see semi-essential, mein arginine and histidine are there. But out of these 2, histidine can be synthesized in adults. So if histidine can be synthesized in adults, it cannot be called as an essential amino acid. So all of the following are essential amino acids except, the answer would be what? Arginine, which is a semi-essential or conditionally essential amino acid. Right? So I hope this was, yes, another question, the enzyme involved in the first committed step of glycolysis. Committed means it is bound to produce, a compound which enters glycolysis only. So it is phosphofructokinase. Always remember, committed enzyme is fructokinase, others are rate limiting enzymes, because it is committed to undergo glycolysis only. So I guess this is all about the discussion of the questions. Thanks so much for listening to my lecture. This is my website. If you have any queries, you can put forward your queries on this website. And if you like my channel, do subscribe to my channel. Thanks a lot. Wishing you all the best for your exam for day after tomorrow. Wishing you luck. Good night.